So we'll get started. Hello, everyone. Welcome everyone to the first SSR Rising Star webinar of 2026. My name is Ewing Chen. I'm a postdoctoral fellow at the National Institute of Environmental Health Sciences. As a member of the Virtual Education Committee of SSR, I have the pleasure to moderate today's webinar with two outstanding speakers, Dr. Cameron Schmidt and Dr. Zhiyuan or ZY Chen for the seminar today. So each speaker will present for about 20 minutes, followed by 10 minutes Q&A. Please submit your questions in the Q&A box, and I'll be moderating those. So our first speaker today, Dr. Cameron Schmidt, who's an assistant professor in the Department of Biology at East Carolina University. Please join me in welcoming Dr. Schmidt for his talk titled, Why Semen Analysis Sucks and What We Can Do About It.
All right. Thanks so much, Ying. Can you see my screen okay?
Yes.
Excellent. Awesome. Sorry, I'll try not to clear my throat too much into the mic. Well, I want to thank you, Ying, for hosting and for that really nice introduction. And I also want to thank the Virtual Education Committee for putting this awesome series on. I'm really flattered to get to be here, and I'm really excited to share some of the work that we've been doing. So my lab is interested in complex systems, and we study how cells use their metabolism to perform actions, how they use information in their environment to compute their physiological states, and how single cells organize into complex tissues and maintain that organization over time. And we do that in a couple of different really interesting systems. So one is the fertilization of eggs by sperm. We're also very interested in signal-related growth and proliferation in cells, primarily in culture. And we're also interested in stable organization of cells within complex tissues. For example, these are seminiferous tubules of the mouse testis, and we're looking at how metabolic enzymes are distributed over the seminiferous tubule. So today I'm going to be talking a little bit more about the practical implications of our work on fertilization, particularly in the context of semen analysis. So what is semen analysis? Well, semen analysis involves assessing the count, motility, and morphology of sperm. And this is done in two primary ways, either by human technicians who can do these measures qualitatively, or by automated systems that can do this in kind of an automatic way. And it's important for a lot of reasons, but the simplest reason is just that men generally want to know whether or not they're fertile, and if they're not, they would like to know what they could do about it. Additionally, it's important because semen functional analysis or sperm functional analyses really form the basis of our understanding of basic male fertility. So for example, in mouse transgenics research, if we're knocking out genes that affect fertility, we might do a mating study and find out that some mice are infertile, but then we need to know why, and often we look at the sperm first. Additionally, it has huge economic impacts for agricultural breeding programs, often where semen is being stored maybe for long periods of time and then shipped out for insemination. And it's particularly important because a lot of males are jerks, and some of those males are 2,000 pounds. So people don't really want to keep them around. And sometimes they're 6,000 pounds and you don't really want to keep them around. Additionally, sometimes they're 4 pounds. But again, sometimes we just don't want to have them around, and we just want to be able to use the semen and also be able to make strong predictions about what it will be able to do. Now, often when I talk about semen analysis, I kind of get this response, well, why do we need to do semen analysis if we can do intracytoplasmic sperm injection, which, as a reminder, just involves selecting a single sperm and then injecting it into an egg to get a fertilized embryo. Well, ICSI is generally expensive. It has relatively low efficiency, and qualitative sperm selection becomes a major problem because you have to choose which sperm you're going to inject. Additionally, ICSI doesn't work really well for many species. So it's an absolutely revolutionary method, but it's definitely not solving all of our male fertility related problems. And in fact, that sperm selection is really important because we've known for several decades now that which sperm fertilizes really does matter, and it can have really dramatic effects on the developing embryo. So, as promised, I thought I should mention why current semen analysis sucks. So the World Health Organization has a standard protocol that it updates every couple of years that highlights how semen analysis should be done. And for fertility classification, we do these count, motility, and morphology assessments, and then compare these to reference tables of large populations of men with self-reported positive fertility status. So then we can use sort of a 95% threshold as a way of flagging fertility. So when certain parameters fall below the 95% threshold, then that may indicate that there's a problem. And then additional functional tests can be applied, but these are usually things that are applied differently in different clinics depending on what they feel is important. And if we have no further explanation, then generally the focus shifts onto female factors to try to explain any fertility issues. So this sucks for several reasons. It can only really diagnose extreme cases. So if there are no sperm or if all the sperm are really messed up in pretty obvious ways. Infertile men are not generally included in the reference tables, and so then it creates some significant confusion about what sperm parameters are important for fertility and which sperm parameters are not. Additionally, it compares the wrong variations. So semen analysis looks at intra-sperm variation, but then the reference tables look at intra-individual variation. It generally doesn't inform the etiology, so it might not really tell you what's wrong with the sperm. And it certainly doesn't inform what you should do about it, even when the infertility causes are relatively clear. Additionally, automated analysis hasn't really been widely incorporated in clinics, despite massive advances in technology in recent decades. And usually human technicians, which are much more expensive, are favored. So I think there's something to learn from the way that other tissues are understood. So if you were to go to the hospital and say that your heart rate was at 120 beats per minute and you feel terrible, and the doctor said, oh, okay, that's terrible. So then they look at a table of 10,000 other people's heart rates who are considered healthy, and they say, well, your heart rate is out of the range, so there must be something wrong with you. That's a pretty unsatisfying diagnosis for a condition like this. And that's basically what we're doing with male fertility. Now, what we can do with the heart, because we have many decades of really, really strong physiological functional theory, is measure parameters that are related to heart function, like heart rate, blood pressure, excitation, contraction coupling, hemodynamics. And then we can relate those specific parameter values with the heart's function as a pump. So if something is wrong, we know exactly how that might affect its pumping physiology and what we can do about it. So how can we improve semen analysis in the same way? We can start to think about semen as a tissue. So here's a really fantastic plot from a study that was looking at rabbits and the semen from rabbits. Rabbits are induced ovulators, so they ovulate when they mate. And what's happening here is this little black bar is kind of showing the viability time space for an ovulated egg. And what we can see is that the acrosome reaction and even the movement functions kind of change for rabbit semen within this time window, implying that there's a kind of collective anticipation of egg availability enacted by the way that the sperm behave. And humans are a little bit more complicated because they're cryptic ovulators and they kind of roll out these small populations of fertilization competent sperm over time. But together, what we can do with that information is kind of imagine that semen is a tissue with this sort of unifying function of finding and fertilizing an egg. And that might seem obvious, but being able to quantify and formalize that searching and finding process in strictly physical terms can help us be able to predict what to do about when semen is not working correctly in the same way that we might be able to do with cardiovascular physiology. So it's important to note that this happens at multiple scales. So this is a single sperm phenomenon, but it's also a many sperm phenomenon. It also happens in multiple dimensions. So what do I mean by that? Well, sperm have to search physical space for an egg, and they do that using their motility. So here's just kind of a single image with tracks of some sperm that were moving. These are just the centroids of the nucleus. Some of those tracks are longer than others, implying that some move faster than others. Some are not moving at all. So this gives us sort of a measure of how sperm search space. But they also have to search molecular space, in the sense that sperm have to undergo this acrosomal exocytosis, which helps them bind to and recognize the egg. And importantly, the way that sperm search physical space, if we were to follow this video out for a longer period of time, we'd see that the sperm actually have these really divergent, complex behaviors that they're exhibiting over time, and that also vary over the population of sperm. And we have no idea how that's related to fertility. And the same is true with the acrosome reaction. So it's been known for decades that if we look at the signaling pathways that control the acrosome reaction, that under capacitating conditions, like in the presence of bicarbonate and calcium, that sperm subpopulations can be observed. So there's this kind of multimodal distribution of different responses. So the sperm don't respond uniformly, and that is likely related to fertility potential. So how should we think about sperm movement? Well, my lab thought quite a bit about this, and over the last couple of years, we've been building computational simulations that are based off of real sperm movement, so we can explore questions in ways that we can't really do with empirical measurements. And one of the things that we found was that if a single sperm searches for an egg, it generally has a hard time finding it. And so it would need to search a lot of space before it finds it. But if we increase the number of sperm, then just by way of having more sperm as a statistical function of a large population, the path to the egg, or the shortest path to the egg, is actually sort of solved by this tissue collective activity. So we've kind of worked on formalizing this and building this into more rigorous physical theory, using random walks and then doing simulations in different kinds of environments. And what we found was that sperm can optimally search areas if they've transitioned between being in a more kind of progressive or linear type motility behavior, or transitioning at some point into a less linear behavior where they spend more time moving locally, implying that sperm can optimally search area when they undergo phase transitions in their movement patterns. And this isn't our data. This is a really beautiful study that was published last year, or as a preprint, by Maisam Zafarani, who's currently a postdoc at Princeton, but he's formerly of Cornell, where he was working on sperm. And what he did here was he sort of trapped a single bovine sperm inside of a microfluidics chamber so that he could watch it over a long period of time. And what he found was that when stimulated with caffeine, the sperm undergoes these phase transitions between these more progressive patterns and these patterns where they're undergoing this kind of circular movement. And then he did some lots of other really wonderful work in that paper, where he was showing how this type of movement pattern can aid a sperm that's kind of randomly searching in a complex environment like the oviductal epithelium. So traditional CASA, or computer-aided semen analysis, relies on phase contrast microscopy and generally just uses these kind of descriptive velocity measures. And then you can classify some basic motility patterns by grouping the observed velocity parameters for a specific sperm. And then in an experimental context, you might measure that in a couple of different groups, and you could say, okay, we've got this percent that are progressive or swimming linearly, this percent that are hyperactive, and that's really been the basis of trying to understand how sperm adaptively change under post-ejaculatory conditions. There's some major challenges with that, even just in data acquisition, though, because phase contrast microscopy has issues with brightness variation among the cells that are moving. The cells are moving very fast. There's signal overlap. This leads to interruptions in tracking, as well as misidentified trajectories. And so in order to get around with this, typically pre-selection is performed, so selecting only certain types of sperm, tracking sperm for very short periods of time, typically less than 1 second, or tracking only very few cells, as well as only tracking the nucleus rather than the whole cell. So we also don't know very much about how flagellar movement corresponds with the actual movement of the sperm for that reason. Now, if we look at the actual tissue, that's a major problem, because all those different things take the measurement of sperm motility completely out of its physiological context. So this is a human semen sample, And there's lots of debris in there. There's tons of cells. They overlap with each other all the time. And interestingly, this is only 0.0005% of the total number of cells that would be in that ejaculated sample. So we really don't have a very good handle on what this tissue is doing. So what can we do? Well, in 2026, the answer to everything is use AI, as I'm sure you've all noted. And in order to do this, what we do is we label some training and test data, and then we would train some sort of an AI model for tracking the sperm, and then we can use a pre-trained model. There are some out there, particularly for cell microscopy. And that solves all our problems. Of course, it's not that easy. There's major challenges to this. Even just the labeling is almost impossible because there's so many cells. It would take someone to manually label this stuff a very, very, very long time. And that would force having really small training data sets that are probably overfit to certain circumstances. Additionally, there's just not very much publicly labeled data out there. And for what's out there, it's not very accurate. So our approach to deal with this has been to develop this kind of closed loop training process where we collect data in order to fit physical models of sperm movement, kind of like the simulations I showed you earlier. And then we can use that as synthetic data in order to pre-train models that can then label real sperm videos that we collect. And then we basically just loop through this whole process iteratively to sort of bootstrap the AI model's ability to track the cells. So here's just kind of an example of what one of these videos looks like. Here's a real sample at 5x magnification at the objective, phase contrast microscopy with just kind of the sperm nuclei is what you can mostly see. And then there's some synthetic data below it, just showing, showing how similar they are. Now, when we used a model like DeepSort, with just a pre-trained model, something that was already trained on other types of microscopy images, we really didn't get very good tracking at all. It shouldn't be blinking like this. It is tracking some of the cells, but those cells have relatively unambiguous tracks. And our link scores, which is just a measure of how good the tracking is, were pretty poor. But then when we use our closed loop data generation method to improve this, and I'll just let this kind of play out so you can see the visual improvement. The tracking was much, much better, and we're able to track it at relatively high densities over much longer time periods than the traditional computer-aided semen analysis. And additionally, just to put a little bit of quantitative data in there, the link score was a really, really dramatic improvement when we followed this method. So what can we do with this? If we can track them, then we can better understand how movement actually corresponds with fertility outcomes. So we need new metrics. So these are the old metrics. These metrics have been around since the 1980s. They're very descriptive. They suffer from issues with time averaging. So these are things like curvilinear velocity and beat cross frequency. And what we're proposing is something that can better capture short time scale movement. So if we zoom in, the sperm kind of wiggles back and forth as it's moving through the media. But then if you zoom out and watch them for longer periods of time, then there's also these long time scale effects where they're either doing things like spiral patterns, or they have some periodicity, or they undergo phase transitions. And all that information is valuable, but we're leaving it on the table. And this is important because we want to be able to connect the actual physiological task of sperm to something that's much more tangible. So we've also been thinking about how search among other types of organisms is actually very well understood from a mathematical sense. So here's the path that an albatross might take while it's searching for fish. And you'll notice that it undergoes these kind of straight runs and then these local periods where it's just moving very locally, searching around, and then it'll take another straight run to a new location and then search around. And it turns out that this is not something unique to albatrosses. This is something that pretty much all foraging animals do. So they'll even spider monkeys will sort of behave like this. And then even on the physical scale of sperm, which is a kind of a microscopic scale, bacteria do something very similar, and the chemical conditions in the environment can modify the way that these short run and straight run distributions affect the search efficiency of the microbes. So I think that my SOM's paper was incredibly important, and maybe one of the most important sperm papers in recent memory, because I think what he's done is shown that these kinds of patterns also apply to sperm, but we have to observe them on the order of minutes, not for less than a second, in order to see these complex patterns. So what can we do with this in the future? Well, I think we need, instead of these kind of standalone CASA systems where you buy a microscope and some proprietary software, we need variable data collection. So there's lots and lots of different ways to view sperm that are out there. That can be done on smartphones. It can be done with microscope attachments. It can be done with many, many different types of microscopes that are in the lab. It can be done with different microscope modalities like holography. And then we can have cloud models that are available to upload data to in order to get better long-term tracking. And in the process of doing that, then those cloud models can also use that data to improve their tracking functions. And additionally, we need to be able to apply some sort of physiological functional theory that connects sperm motility with search. So I also mentioned that sperm have extensive heterogeneity, and this relates to their ability to search other kinds of spaces other than just physical space, like molecular space. So we've known for many decades that sperm capacitation, which involves the acrosome reaction as well as changes in motility pattern, are tightly regulated by cell signaling pathways, and that these responses are heterogeneous. But we don't really know what to do with it. So my lab's been thinking a lot about this, and we've been thinking about how we can imagine a sperm population as sort of a phenotype manifold. So we've got 3 distinct sperm here. Those sperm consist of whatever traits that we can measure about them. And so if we can measure those traits uniformly, then we can sort of create each a little vector that represents what that sperm is at any given moment in time. And we can use flow cytometry to measure this, and it has great single cell resolution, and we have the potential in some cases to even sort cells based off of specific traits. So we developed a 2D culture system so that we could scale different inputs that might determine the way the sperm behaves. So either changing input signals like calcium or bicarbonate or progesterone, something like that, or just looking at how sperm populations evolve over time. And I realize that this heat map is a little bit busy, but it's just showing you that different conditions can cause sperm calcium measurements, intracellular calcium measurements, to kind of change differentially. So the phenotype manifold that we're going to look at today involves measuring intracellular calcium, live dead status, as well as the acrosome reaction. This is all in live cells. And these are done primarily in mouse cauda epididymal sperm, because we also want to be able to relate this to IVF, which is something that we're not able to do for human sperm in my lab. So the problem with most approaches of using flow cytometry, because we're certainly not the first people to use flow cytometry to analyze sperm, is that the way it's approached often involves kind of like taking all this big data and then compressing it into what I would call small data. So we biologists tend to kind of measure everything and then reduce the dimensionality as much as we can. I'm sure we're all familiar with that from RNA-seq experiments. And then we calculate the averages or means, and then we try to figure out how to kind of cram that into a hypothesis test. And the reason is because stochastic modeling just isn't really widely adopted in the field of biology, at least not as much as it probably could be. So when we're thinking about stochastic modeling for this problem, we wanted to be able to define clear subpopulations. So we have live unreacted cells, live reacted cells, where reacted refers to the acrosome reaction, and then dead reacted and dead unreacted. And those are just things that we can measure with these particular dyes, but we could probably measure other things as well in the future just using different dyes. So in order to classify the cells that we're measuring as belonging to one or more of these, one or the other of these different subpopulations, we developed a kernel density estimate Bayes classifier that basically just uses positive or negative experimental controls, like treatment with a calcium ionophore or treatment with a detergent to kill the cells, to create probability distributions for the kinds of signals that we might see under known conditions, so that when we look at cells that are measured experimentally later, we can say, okay, do they fall in this bin or do they fall in that bin? And that's basically how we classify them. So what can we do with this? What can we predict? So we had this really kind of surprising inspiration from some papers that were focused on the gut microbiome, and trying to understand compositional analysis in those populations, how those populations change over time, especially when they have these really complex subpopulations. So what the statistics that we're using does is, if you imagine you had a coin toss, and you could say, like, how many times am I going to see heads if I flip this coin 10 times? Then you could use the binomial distribution to predict what the probability of that would be. And the same is true for things that have more than just two outcomes, like a pair of dice. So how many times am I going to see 2 6s if I roll these dice 10 times? Something along those lines. Now, the reason that, or the feature of these two systems that's really useful, is that we know what the underlying probabilities are. We know that the probability of a coin toss is 1/2. We know that the probability of rolling a 6 is 1 out of 6, for example. So we developed statistics that can look at real data, and then the goal of these types of studies, or sorry, the goal of these statistics is to actually estimate the underlying probabilities. So it'd be sort of like seeing a bunch of die rolls and then trying to estimate what the probability of observing one of those combinations of faces actually is. So here we're just looking at different subpopulations under increasing concentrations of calcium or increasing concentrations of bicarbonate. The thing to take away from this is that in the live reacted cell population, which is the cell population that's probably has the greatest fertility potential, the conditions are actually very different than what typical IVF conditions are. And then we can estimate the underlying probabilities using those statistics, and we're testing the ability to use those probabilities in the future to make inference about the ideal conditions for doing IVF. All right, I'll try to move really quick. I see that I'm getting pretty close to time. So in terms of the bigger picture, we now want to be able to look at other factors like how do the cell populations change over time? What happens if we change the signals midway through? What happens if we give them multiple signals? Is this like a uniform distribution of different responses, or is there a population gain in different cells being able to respond to different signals at different times? Ultimately, the goal is to improve IVF efficiency, but in order to do that, we're not just going to incubate fixed densities of cells, which is the typical practice in IVF, but instead we're going to try to use those statistics to predict how many cells we should be incubating with the eggs and for how long, based on the proportions of fertility competent cells that we would predict are there. And then often one of the kind of pieces of criticism I get about this is that, well, flow cytometry is not widely adopted in the clinic, and it's unlikely to be, and I totally agree with that. But there are definitely technologies out there like microfluidic flow cytometry that are cheap and totally accessible and just need to be sort of tailored to those specific applications. So the technology is there, but what we need to do is make sure that those measurements are actually meaningful in some way, and then I don't doubt that they'll be incorporated. So in terms of takeaways, the things that we know are that motility corresponds with fertility, but we don't really know how that motility corresponds with fertility. And for that, we need a much more rigorous functional theory of fertilization. We also know that capacitation is tightly regulated by signaling factors, but we don't really know how underlying heterogeneity informs the fertility competence. So what we need to overcome this is we need new tools, we need new metrics, and we need more than anything else, a functional theory of sperm physiology as well as sperm selection. So thank you all so much for listening. I want to I have some really fantastic students in the lab that all contributed to this research. I want to make a special thanks to Chris Geyer, who's been incredibly supportive as I've been kind of transitioning into reproductive biology, as well as Chip Muller, who's out at University of Washington, who's really, really helpful in advising us about how to do human sperm capacitation. And then the very last thing I want to say, I realize I'm like right at time, is that SSR has a podcast, and it's awesome. We do interviews with award-winning reproductive scientists, interviews with active researchers, lots of career advice, cutting-edge science. You can get it at ssr.org or wherever you get your podcasts.
Thank you, Cam, for a very insightful talk. So we'll take one question Could a deep sort CLT model be trained to distinguish between various mechanisms of action in novel contraceptives, such as Caspar channel inhibition or adenyl cyclase inhibition, by analyzing specific changes in sperm trajectory and capacitation patterns post-ejaculation?
I think that's certainly possible. I think the kinds of metrics that we use to analyze motility now are like we might be able to compare different effects on those motility parameters, but those motility parameters are flawed for a number of different reasons. So having a better sense of the variation and how that variation in motility relates to the actual physiological task is something that I think we need a little bit more information for. But I think it's absolutely possible that we could miss some of the subtle effects of things like contraceptive agents that maybe they don't have an apparent effect on normal CASA parameters that we use now, but do have an effect on things like phase transitions between or phase transition rate between progressive or less progressive motility.
Thank you. If you have more questions, please feel free to reach out directly to Dr. Schmidt, and we will now move on to the second speaker. Our second speaker is Dr. Zhiyuan, or ZY Chen, who's an assistant professor in the Division of Developmental Biology and the Reproductive Sciences Center at Cincinnati Children's Hospital. His seminar is titled Polycomb-Repressive Debuquidinase Complex Safeguards Oocyte Epigenome and Female Fertility by Restraining Polycomb Activity. Please join me in welcoming Dr. Chen.
Thank you, Ian, for the introduction, and thank the organizers for giving me this opportunity to talk today. Today, I like to share some of our recent progresses in understanding oocyte epigenome and female fertility. Dr. Schmitz gave a wonderful talk about how we should improve the semen analysis, and I think we all agree that healthy oocyte is also crucial for successful fertilization and early development. Defects in oocyte and early embryo competence can manifest as a range of phenotypes, such as empty follicles, maturation arrest, fertilization failure, zygote arrest, or early embryonic arrest, or mixed phenotypes across IVF cycles. Some of these OECD defects can be linked to established genes. For example, in this paper published last year, in their cohort, they found that patients with empty follicles, about 50% of these can be associated with mutations in ZP, Polusta protein ZP123. However, you can also realize that there's a substantial fraction of these patients, they do not have identifiable genetic contributions, especially for those involving early embryonic arrests. So of these, about 1,000 patients with early embryonic arrests, only about 78 of those can be genetically diagnosed. So these data indicate that we still need to better understand the molecular mechanisms involved in oogenesis and early embryo development. So in our lab, we are broadly interested in molecular mechanisms in oogenesis and early development, particularly focusing on transcription and chromatin mechanisms. So during postnatal oocyte growth, the oocyte already arrest at prophase 1, so they just dramatically increase the cell volume without cell division. And throughout this process, the oocyte accumulate maternal RNA and proteins that are essential for the subsequent meiotic maturation and early embryo development. So the global transcription is shut down before ovulation and only resumes after fertilization when the zygotic genome activation, or ZGA, takes place. So in the mouse, there are two waves of ZGA. The minor waves happens at late 1 cell stage, and major wave completes at late 2 cell stage. During ZGA, there's a subset of retrotransposons that are transiently activated. And one of our lab's direction is to understand regulations and functions of these totipotency-associated retrotransposons. We're also interested in understanding chromatin dynamics and functions during these processes because chromatin modifications, chromatin variants are key regulators of transcription, and they also undergo substantial changes during these developmental windows. For example, histone 3 lysine 27 acylation is a well-known active chromatin marker for promoters and enhancers, and the global K27 acylation progressively increase during the oocyte growth and then undergo a few rounds of resettings or reprogramming during maturation and early embryo development. So today I will talk about some of our recent understanding of how ketone acetylation is regulated during oogenesis. So to better define the ketone acetyl dynamics, we manually separated oocytes based on their diameters into 4 groups, and then profiled ketone acetylation. So as we can see from this genome browser view, I hope you can appreciate that there's a global increase of the ketone acetylation when the oocyte size increase. In particular, during the final stage of oocyte growth, from growing oocyte 2 to following growth oocyte, we can see a prevalent or widespread increase of ketone acetylation. It's in particular surprising or unexpected is that even in gene poor regions, we also see such widespread increase of ketone acetylation, because gene poor regions usually are heterochromatin, and you wouldn't expect many or a lot of the active chromatin marks like ketone acetylation. So these data actually in well agreement with the recent star chip data, ketone acetylation star chip data from Wei Xi's lab at Tsinghua University. They also show in their paper that fully grown oocytes have a substantial higher level of ketone acetylation in gene poor regions compared to growing oocytes collected from postnatal day 7 or day 10 mice. So these data really raise an intriguing question that has motivated our work to answer what's the underlying mechanism and functional significance of prevalent ketone acetylation in fully grown oocytes. So in fully grown oocytes, ketone acetylation is mutually exclusive polycomb repressive markers, including H2A ubiquitin and ketone 7 triamylation. So these two repressive markers are deposited by large chromatin complexes called polycomb repressive complex 1 and PRC2. PRC1 has a E3 ubiquitin ligase activity and deposits monoubiquitin to the histone H2A, and the PRC2 deposits or catalyzes monodiene trimethylation on histone 3 lysine 27. So previously, we and others have demonstrated that polycomb function regulates in oogenesis, regulates a number of processes depending on which subunit is deleted or what cree used. We have known that polycomb function can regulate variant reserve, oocyte developmental competence, 3D chromatin, non-canonical imprinting, and imprinted axon activation. Despite these established roles, I think one major knowledge gap we try to address in this work is that how is polycomb activity is restricted to avoid heterochromatin spreading into euchromatin regions, particularly those marked by ketone acetylation. So in the work I'm going to share with you, basically what we found is that we identify polycomb repressive deubiquinase, or PRDUB, is a key negative regulator of polycomb activity during oogenesis. So PRDUB consists of the BAP1, which is a ubiquitin hydrolase that can specifically remove ubiquitin from histone H2A. It also contains an essential cofactor called ASXL familase proteins. So in a fully grown GV oocyte, the polycomb repressive markers like H2A ubiquitin and ketone 7 trimethylation, they are highly colocalized because of the extensive crosstalk between PRC1 and PRC2. They are mutually exclusive with the ketone 7 acetylation, which is an active mark. What we found is that loss of BAP1 in oocyte causes a widespread increase of H2A ubiquitin, although this increase is much less, to a less extent, at highly active oocyte. And we also found that the BAP1 primarily contributes to gene activation and dispensable for the polycomb mediated silencing. For those BAP1 dependent genes, we found that there's a ectopic gain of ketone 7 trimethylation and a corresponding loss of ketone 7 acetylation. So functionally, we learned that BAP1 is essential for the oocyte developmental competence. BAP1 non-oocytes cannot properly support maternal to zygotic transition, pre-implantation development, and the loss of maternal BAP1 severely impaired female fertility. So, so during the rest of my talk, I will walk you through with the experimental evidence that support these major conclusions. I'd also like to highlight that this work was mostly carried out by Jinwen in the lab, and this work was on bioRxiv last year and just accepted this week. So in this story, we first set out to identify key histone H2A deubiquinase or H2A DUBs in oocytes and zygotes. So we used the candidate approach. In this design, we micro-injected individual H2A-DUB mRNAs into early zygote and collect late zygote for immunostaining. We selected the 5 candidates, USP16, USP28, USP21, MYSM1, and BAP1, because they are reported H2A-DUB activity and also their high expression levels in oocytes and zygotes. So as we can see from this immunostaining, USP21 overexpression can efficiently remove H2A ubiquitin within 6 hours. BAP1 co-injection with ASXL1, but not BAP1 injection alone, also erases H2A ubiquitin in zygote. For other 3 candidates, USP16 and 28 and M1SM1, they have little effect on H2A ubiquitin. We should note that these 3 enzymes, they may function as H2A-DUBs at other developmental stages or require additional cofactors in zygotes. Because USP21 has been reported before that USP21 knockout mice are viable and fertile, so we decided to focus on BAP1 for downstream follow-up loss of function analysis. So we use a CRISPR-Cas9 system to conditionally delete BAP1 in growing in oocytes because constitutive knockout BAP1 is embryonic lethal. So we use a gene-derived CRE, which is starting to express on postnatal day 3 in primordial follicles. At 6 to 9 weeks of age, based on ovarian histology, we didn't see much different abnormalities. And also, we were able to collect comparable number of oocytes, fully grown oocytes from both from the conditional knockout female mice. So these data suggest that the loss of BAP1 may not affect follicular genesis. Consistent with the function that BAP1 is a H2A ubiquitinase, so we saw an increase of H2A ubiquitin level by immunostaining in fully grown oocytes. Because immunostaining can only give us a global view, so we thought to investigate what genomic regions are affected in fully grown oocytes by BAP1 knockout. So to address this question, we performed SMARCI normalized cut and run in control and conditional knockout. So we can see here that the 2 control replicates are highly correlated with each other and also match well with our previously published data. However, when we see the conditional knockout, we see a global, pervasive, or a widespread increase of H2A ubiquitin. To validate further, we calculated log2 fold change of H2A ubiquitin at different genetic elements, including putative enhancers, promoters, gene bodies, and intergenic regions. We saw an increase of H2A ubiquitin across all the genetic elements, and this increase in oocytes seems more pronounced than in mouse embryonic stem cells. So because we know that H2A ubiquitin can guide PRC2 to deposit K27 trimethylation, and K27 trimethylation is mutually exclusive with K27 acylation, so we next ask how BAP1 knockout may affect these 2 histone modifications. So unlike H2A ubiquitin, which is widely spread, broadly increased in conditional knockout oocytes, we found region-specific increase or gain of K27 trimethylation, for example, in this area. And this increase of K27 trimethylation is associated with or correlated with the loss of K27 acylation. We also noticed that these changes preferentially occur in gene-poor regions versus gene-rich regions. So to validate genome-wide, we identified about 9,000 regions that lost K27 acylation in a conditional knockout, and we see a K27 acylation loss is globally associated with the K27 trimethyl gain. We also observed that in H2A more ubiquitin is increased similarly in both K27 acylation loss and K27 acetyl no change regions. This indicating that K27 ubiquitin increase alone cannot explain why certain regions are affected but not other regions. For those K27 acetyl loss regions, we saw it's enriched in the intergenic regions compared to those no change regions, suggesting a role of BAP1 in regulating enhancer activities. We also checked the gene expression changes. Basically, we found that those genes associated with promoter or gene body K27 acylation loss, they are downregulated in the conditional knockout oocytes. So one intriguing question from this analysis is that why some regions are more sensitive to BAP1 knockout than others. From this heat map, we can see that increase in H2A ubiquitin is not the sole determining factor. So to explore this further, we collaborated with Satoshi Namikawa's lab at UC Davis, and to collect it and analyze the postnatal day 1 non-growing oocytes. So this stage is before the GDM9 reactivation. And we also reanalyzed public data at P7, P10 growing oocytes. So this is the genome browser view at the FGF10 locus. We can see that some of these keto peaks, they are already present in the non-growing oocytes, and these peaks seem to be more resistant or less sensitive to BAP1 loss. However, for some other regions like here, these regions only gave keto isolation after P10 growing oocytes, and then these regions lost keto isolation upon BAP1 loss. So from this data, we think what happens is that BAP1 is critical to establish keto isolation during postnatal oocyte growth, but is dispensable for maintenance of pre-existing keto marks. So to summarize the first part of my talk, basically we showed that BAP1 limits pervasive H3 ubiquitin in accumulation in oocytes. It also protects keto isolation from ectopic keto trimethyl gain. And we show that BAP1 is essential for gene activation in subset oocyte, preferentially in gene poor regions. So this actually somewhat kind of contradict with previous data because BAP1 has been shown as a gene repressor in Drosophila. Actually, that's how the name comes from, polyclonal repressive. So in Drosophila, loss of BAP1 causes derepression polyclonal targets, including Hox genes such as UBX. And this derepression can be rescued by a transgene, BAP1 transgene, but not a catalytic dead BAP1 form. So in mouse embryonic stem cells, BAP1 is also implicated in polyclonal silencing. What people believe is that widespread increase of H3 ubiquitin in BAP1 knockout ES cells causes titrate away this PRC1, PRC2 complex, so causes derepression of these genes. So what happens in the mouse oocytes? So to address this question, we analyzed 1,700 oocyte polyclonal targets defined by the enrichment of keto-7 trimethylation. As a side-by-side comparison, we also reanalyzed the RNA data from PRC1 and PRC2 knockout oocytes. These were driven by ZB3 Cree or GDM9 Cree. Both Crees are activated like postnatally around P3 and P5. So these are one diagram showing the derepressed oocyte polyclonal targets in different mutants. As we can see, the PRC1 plays a more predominant role in regulating or mediating polyclonal silencing than PRC2. In contrast, BAP1 knockout only causes about 7 polyclonal targets derepressed. Furthermore, the magnitude of derepression is much mild in BAP1 knockout when compared to PRC1 and PRC2 knockout. So when we look at global gene expression changes, not just polyclonal targets, we found that PRC1 and PRC2 knockout causes more genes upregulated than downregulated. This is actually an opposite pattern. In BAP1 knockout, we actually see more downregulated genes than upregulated genes. And for these downregulated genes, they are enriched in pathways like cell adhesion, including CDH and DSC2, and also growth factor binding, such as FGF10 and EGFR. So for summary, to summarize the second part of the story, I hope that our data is convincing to show that BAP1 primarily contributes to gene activation and is largely dispensable for polyclonal-mediated silencing. I think the reason why this is because the PRC1 and PRC2, they're highly expressed in oocytes than the ES cells, even though the widespread increase of H3 ubiquitin may titrate away some of these complexes, but still they're enough to maintain the polyclonal silencing. So with all these chromatin changes and gene expression changes, we are interested in ask what's the developmental consequence of these BAP1 non-oocytes. So to evaluate developmental competence of BAP1 knockout oocytes, we cross the control and conditional knockout female mice with wild-type E6 mice. So in this breeding scheme, the embryos, they do not have the maternal BAP1, but the paternal copy still is wild-type. So these are maternal knockout embryos. At E3.5 expected control group, we saw most embryos reach to blastocyst stage. However, in the maternal knockout group, we saw most embryos arrested in morula, or maybe some embryos even more poor morphology. It was surprising to us that when we cultured these embryos for additional 24 hours, we saw many of these actually can reach to blastocyst stage. However, their sizes are much smaller than control. So we also performed immunostaining to analyze the cell counts and the lineage marker expressions. We found that the maternal knockout embryos show much less cell numbers compared to control, and also they have defective expression of leading markers such as GATA4 for the primitive endoderm, NANOG for epiblast, and CDX2 for the trophectoderm. So since the maternal knockout still have a one wild-type copy, so we next want to tease apart whether this defect is a maternal effect or due to haploinsufficiency. So to address this question, we further cross the control female mice with the PIM-3 BAP1 flox mice. So PIM-3 protein 3 is expressed at a very late stage of spermatogenesis, and so this breeding scheme generates paternal knockout embryos. We also generate maternal and zygotic knockout embryos by crossing GDM3 and PIM-3 BAP1 flox mice. So what we saw from these IVF experiments is that the paternal knockout actually has no effect on the preimplantation development because these embryos can reach to blastocysts and with normal morphology. And this means that the maternal knockout phenotype is due to the maternal effect, but not the haploinsufficiency. In addition, the maternal zygotic knockout actually has stronger phenotype, indicating that the paternal copy may somewhat rescue the defect we observed in the maternal knockout. When we check the developmental progression, we found that the developmental delay or arrest starts around 2-cell to 4-cell transition. And this is a critical window when the maternal to zygotic transition takes place. Basically, maternal RNA need to be degraded and ZJ need to be ZGJ need to be activated. So we next check whether maternal to zygotic transition is normal in BAP1 maternal knockout embryos. So to address this question, we collected late 1-cell, early 2-cell, late 2-cell embryos for total RNA-seq analysis. And the minor ZGA starts around the late 1-cell and completes at early 2-cell, and the major ZGA completes at late 2-cell stage. So based on this analysis, we found that about 20 to 30% of the ZGA genes, minor or major ZGA genes, are downregulated, more than twofold change in the maternal knockout groups. In addition, at late 2-cell stage, we saw about 13% of maternal genes are upregulated, suggesting a defect in maternal RNA clearance. These abnormal ZGA genes or maternal genes include those well known to be important for preimplantation development, such as CDK2, AP1, R5A2, O-box proteins, including zygotic O-box and maternal O-box proteins, as well as some maternal genes that are known to be important for early embryo development. So we think that these abnormal MZT may contribute to defective preimplantation development and blastocyst formation. So we next want to ask, what's the developmental potential of these low-quality BAP1 maternal knockout and blastocysts, whether they can still implant and develop to term. So we first checked E6.5 about 2 days after implantation. We saw a reduced number of implantation sites, as well as a reduced number of embryos can be recovered from the uterus. And even for those embryos recovered, we saw a much smaller size compared to the control embryos. I think this data is consistent with or match well with the E18.5 data. We saw reduced litter size, live pups at this stage, and we noticed many embryos many resorption sites at E18.5. So for 6 months breeding trial, we noticed that loss of maternal BAP1 severely impairs the female fertility due to both reduced litter sizes as well as the litter numbers. So to summarize the third part of my talk, I hope that our data are convincing to show that BAP1-dependent transcription activation in oocytes is essential for oocyte developmental competence. Loss of BAP1 causes abnormal maternal to zygotic transition, defective preimplantation development, and without BAP1, loss of BAP1 severely impairs female fertility. So in this story, we also explored the intergenerational inheritance of kidney-7 translation as well as imprinting, but due to time limit, I'm not going to expand these topics. Welcome to check our bioarchive, or the revised manuscript should be online soon. So currently, we are still addressing some interesting follow-up questions. The first question we will try to answer is, how does BAP1 protect kidney translation in oocytes? We think this could happen in both indirect and direct mechanisms. So indirectly, BAP1 loss may lead to ectopic kidney-7 trimethylation gain, which in turn causes reduction of kidney-7 methylation. And we think there should be also a non-mutually exclusive possibility is that BAP1 loss may directly regulate kidney-7 methylation because the ASXL protein in the PRDB complex has been reported to directly interact with enhancer machinery such as BRD4. So we are testing these two possibilities right now. Another intriguing question is that we noticed that these early killing peaks in P1 non-growing oocyte, they are more resistant to BAP1 loss compared to other regions. It is due to these regions are intrinsically independent of BAP1 or simply due to the timing of GDNAC reactivation after P1. So we're currently addressing whether BAP1 may regulate killing isolation dynamics and transcription during perinatal oocyte transition. So for acknowledgement, I really want to thank my current and former members, especially for Jinwen, who has been the leading person on this project. And also I'd like to thank our collaborators, Satoshi and Menwen, for their generous help and support to collect and analyze non-growing and growing oocytes. And our funding source comes from Cincinnati Children's and NIH. So that will be all, and I'd like to take any questions.
Thank you. Thank you for an exciting talk and congratulations on the publication. We'll now open up for questions. Please put your questions in the Q&A box. And I guess I can start with the first one. How is BAP1 activity being regulated? Do you have any thought? And what's regulating its specificity to target gene pool region?
Yeah, that's a very good question. So that's still something we are working on right now. It seems like BAP1 has a widespread activity across genome based on the pervasive increase of H2A ubiquitin. But when you do ChIP-seq, you also see that BAP1 peaks, especially localized some enhancer and promoter regions. So seems like right now my understanding is that there's a ubiquitous activity of BAP1. Seems like no specific targeting mechanism, but also there is a mechanism can specifically recruit BAP1 to certain places. So that's something that we are still working on trying to figure out.
Thank you. Okay, we have another question. Do you know if the overall level of H2A ubiquitination level is higher in oocytes when compared to ES cells? Curious because it seems like there is no dilution of repressive factors in oocytes, as you mentioned.
Yes, so that's a very good question. So I think there are maybe a couple of reasons why that what do we see here, why the increase of H2 ubiquitin is more pronounced in oocytes than ES cells. One reason is that for the ES cell data, that's an inducible knockout. So they analyze the cells after tamoxifen induction 3 days later. But for oocytes, we started GDM9 started to express around postnatal T3 in primordial follicles. So basically a few weeks for allowed oocyte to accumulate this histone modification. And again, it's right that oocyte does not divide during this time, so there's no dilution mechanism. So I think both may be big due to one is the timing, longer time effect. Another is that there's no division, cell division mediate dilution. So that's why I think that the oocyte has more H2A ubiquitin increase in the BAP1 knockout mice.
How do you explain the difference in activity of BAP1 in the mouse model versus Drosophila? Does the Drosophila form also interact with the ASXL homolog?
Oh yeah, that's a tough question. I think that's the field that's actually the field is trying to understand why it's so different. Drosophila indeed has a similar HASXL homolog, but it seems like the Drosophila basically you knock out BAP1 also causes a widespread increase of H2 ubiquitin. But the people believe is that in Drosophila, without the increase of H2 ubiquitin actually disrupts polycomb-mediated genome compaction, which is actually somehow we don't see here in our data or in our system. So it seems like BAP1's function has a context-specific manner. So it depends on really what cell context or what organism you are studying. In regarding why Drosophila and mouse oocytes are so different, we still don't know the answer yet.
We'll take one last question. Is it known if transcription factors or RNA degradation enzyme can be ubiquitinated? Could that partly explain the increase in maternal transcript abundance and decrease in zygotic ones?
Yeah, so most reports have reported that BAP1 mainly deubiquitinates histone H2A, but there are some reports say BAP1 may also remove ubiquitin from non-histone proteins. In that, we haven't addressed that in this work. I think to address that, we need to do some proteomics studies to identify potential direct non-histone targets for BAP1. So that's a possibility we have not tested yet.
Okay, thank you. As we're at the top of the hour, if you have more questions, please feel free to direct them directly to either speakers. And before we wrap up, I'd like to congratulate both Dr. Schmidt and Dr. Chen again for the Rising Star Award. Thank you all for joining us today, and we look forward to seeing you at our next Rising Star webinar on March 11th, featuring Dr. Fernesa Morgan Iliera and Dr. Kara McKinley. Thank you. Bye.